Any other questions? Okay, so a few minor topics that are probably important background genetics-wise for EvoDevo. So things especially pertinent to today's class in this paper. Ploidy. Does anybody want to give a brief definition of ploidy? I'm only asking because I don't want to talk for the whole remaining hour. It's the number of, it's the number of sets of chromosomes. So in other words, we're talking about mice today. So this is a mouse karyogram. This is a photograph of all of the chromosomes in one mouse cell. This is the full set of DNA that they have, excluding the mitochondrial DNA, which we don't need to worry about. So when you look at this, there are 20 different chromosomes, numbered 1 through 19, and then the 20th set are the sex chromosomes. They're not numbered, but that's the 20th pair. And the ploidy then is pair. Because there are 2 of every chromosome up here, there are 2 copies of chromosome 1, there are 2 copies of chromosome 2, there are 2 copies of chromosome 3. The ploidy is 2. There are 2 sets of every chromosome. And in genetics, 2 ploidy is called diploidy, diploidy, or 2 ploidy. So mice are diploid, humans are diploid. We also have 2 copies. Most mammals are diploid, have 2 copies of every chromosome. The reason this is important for EvoDevo, hopefully if you reflect on what we ended class with last time, we were talking about duplicate genes and what happens to them. You've got an extra cell phone, you can use it as a doorstop. That's neo-functionalization. If your cell phone copy breaks, one of them is still useful as a phone, the other one is a computer. That's sub-functionalization. You still have the same functions are able to take place just with 2 different cell phones or genes instead of 1, and so forth. So each of us and each mouse then has 2 copies of every gene because they're diploid. So that's the natural situation for every organism, mouse, human. Anyway, so we already come with this sort of built-in gene, extra gene. That's before we talk about gene duplication. That's just sort of wild type, that's normal. We all have 2 copies of every gene. And the reason that that's important for this paper is because then we have to talk about dominance. So all of our proteins, most of our proteins that are in our cells, like transcription factors, like we talked about Monday, will either be dominant or recessive. And we'll talk more about that as the semester goes on. But the point related to this paper specifically is if you're a transcription factor, what is your job? What's your function? What does that protein do?
Bring all of the proteins.
So it changes gene expression.
Bring all the other proteins in to express a gene.
It helps grab other proteins that are useful for transcription and translation, making the gene making an RNA from the gene, then making a protein based on the information in the RNA molecule. But what is the protein physically doing to do all of that? It's binding DNA. The transcription factor physically binds the DNA. So, if you're diploid, you've got 2 chromosomes, each of which have 1 copy of this transcription factor. So again, the arrows indicate the positions of genes. So this one gene is the 2 different copies. 1 chromosome you get from your mom at fertilization, the other one from your dad, maternal and paternal. If you're diploid, you get 1 copy, 1 set of chromosomes from your mom, 1 set from your dad. What happens, and this is where dominance and recessiveness comes in. What's going to happen, do you think, what's your prediction, if the copy of this transcription factor, that gene that you got from your mom, has a mutation in it so it doesn't make a functional protein? What happens to your transcription? What happens to you in development if one of the 2 transcription factor copies that you have doesn't work?
The protein that was supposed to be expressed doesn't get expressed. The gene.
Okay, so the protein maybe doesn't get expressed, but what about
The other
dad's copy of?
probably will compensate for the missing ones or replicate.
That is exactly the definition. That's how geneticists, molecular biologists, and developmental biologists define, that's one way we define dominance. So if you are missing one copy of a gene, but the other copy can do the job fine by itself, if it's wild type, if it's normal, if it doesn't have any mutations, if this gene is still producing these black proteins that go bind to the DNA and turn on transcription, you're fine. The main caveat here is that there are some cases, which could be including transcription factors, where one copy of the gene makes half as much protein. So you can imagine this scenario. Remember we talked last time about protein concentration and how high concentration of protein makes it easy for those transcription factors to bind the DNA sequence they're meant to bind and turn on transcription. So that is sort of a gray area. If you've got one copy of the gene instead of two, maybe the amount of protein is half as much. That might have an effect on development. Just from that simple story, it's hard to draw a conclusion about what the outcome would be. But in general, I think it's a safe starting point to assume that if you don't have one copy of a gene, your other copy is wild type, it doesn't have any mutations, it's okay, that it will still produce the normal protein, and let's say most of the time, having half as much protein is fine. Because that normal looking protein can still bind the DNA sequence, it can still cause those genes to turn on. Having extra copies of genes is often good. Having half as many as you normally do, sometimes bad, sometimes not. That's actually an outstanding question in the field of development. One other thing I wish I had told you before you looked at this paper, but we'll talk about it. It may have impacted your ability to understand what was going on or not, is that in a diploid organism, you've got two copies of chromosome 1, although this is not where the Hox genes are. Let's say you have that wild type version of the gene HOX1 from your dad. So let's say that's one paternal. That's not actually formal genetic notation, that's my notation, the parenthesis P at the end. But just for the sake of this story. And then let's say you've got a mutation in the maternal version like we were just talking about. So I'm going to just write that one in red. That's the maternal copy. So we're going to see this notation a lot this semester, which is why it's important to get it out of the way now. A geneticist will abbreviate that drawing in a particular way. So how would a geneticist write the genotype, which is just a alphanumerical code that tells us this picture, basically? The genotype on one chromosome is listed on one line, and then there's a slash, which indicates the separation of the two different pieces of DNA, the two different chromosomes, and then the other genotype is written on the other side of the slash.
And big H and small h for
So
dominance.
you could do big H, capital H for the wild type gene. That's a common genetic standard, that a wild type version of a gene be a capital letter, and a recessive or a mutant either would be lowercase. So this notation then would indicate that an individual, say a mouse, has that genotype, capital black H, whatever notation the scientists that wrote the paper are using, on one chromosome, and the slash will indicate what's happening on the other copy of the same chromosome. These scientists today actually broke that nomenclature rule. They didn't use the slash, you might have noticed. But this we'll see in a lot of papers to indicate genotypes, because there's going to be a lot of messing with the genotypes, the DNA of organisms, and seeing how that affects their development. Any questions or concerns about that? Okay, One other point, let's just test your understanding in a non-point bearing manner. There are two other combinations then of genotypes. You could have either inherited two wild type copies of this gene, one from mom, one from dad. So you'd be capital H / capital H, right? What do we call that, geneticists, when the two versions of the gene are identical? It's called homozygous. Same homo. Zygous has to do with the zygote, or the fact that that means that those two same genes were in both of the gametes, in the sperm and in the egg. So homozygous means the sperm and the egg had the same version of that gene. So this is homozygous wild type. Another possibility is that you get from two parents two mutant copies, which we would call homozygous mutant, I guess. Oops, pen. A-N-T. I can't even fit an N in there. That says mutant. It doesn't look like that. It looks like hieroglyphics. What's the third possible combination of genes that an individual with parents that have big H's and little h's can get? This is called heterozygous. Hetero, different, means each of the gametes carried a different version of the gene, and that would be symbolized by capital / lowercase. So that's the situation we were just talking about when I was asking about, is the heterozygote okay? If you've got one mutant version and one wild type version of the gene, what do you predict will happen? And so the real question in developmental biology in general, and especially Evo-Devo, then is, this is the sort of, if you're taking notes at all, write this down, sort of a point. You don't have to, but if you want to. A question often in developmental biology is, what is the phenotype, or what is the trait? What does this organism that's a heterozygote look like? We're pretty sure that if you only have mutant versions of genes, you're going to have, if you have a problem with development, this is the individual that's going to show that problem. No normal versions of the gene. It's all mutant versions of the gene. This individual is going to be our positive control, our reference. This is the normally developing individual. We're pretty sure that if anything happens, this individual is going to be screwed up. But the way we learn most about developmental genetics is looking at the heterozygotes. Is having one copy of the wild type gene enough for normal development to occur, or is it not enough? That is this question mark going to be wild type? My bias. Is the mouse going to look normal, or is it going to look mutant? Or is it going to look like neither of those two? Is it something in between? So that's a pattern that would be useful to think about every time we read a developmental genetics paper. It's always a big question looming in the minds of the geneticists. And I think this is the last background bit, which is good because we can get at least going on this paper. If you think you have your favorite gene and you want to know what it does in development, what sort of things, not necessarily knowing anything about how in the laboratory genetics works, what sort of things would you want to try to do to a gene to know what it does to an organism in development?
No account for overexpress.
Okay, so you could do change expression. So that's something that we're going to commonly see. So what sorts of things, my finger doesn't work to make dots anymore. Very sad. Okay, change expression. Awesome.
Oh, sorry, I was gonna respond to the question.
Go for it.
Oh, okay. You can isolate the mutants of interest, like unusual appearances.
Yep.
And then proceed towards identifying the genes or gene responsible for the observed phenotype.
Yes, so genetic mapping. But once you find that gene, then what do you do with that gene?
You can either study the mutant that lacks the gene,
Okay.
Or express an altered version of the gene.
Yep. Okay, so we've got, if you think a gene has a role in development, you can get rid of the gene. You can either make it make less protein or no protein. That's one way to change its expression. And then you just look at the animal and you say, does it look like it develops differently than a gene or an animal that has a normal level of that gene or protein? Or you could, instead of underexpress or totally knock out, just get rid of the gene, you can overexpress. Okay. What else? There's one other main way that geneticists will test the role of genes. So this approach, changing the expression of a gene or getting rid of it, is colloquially called the lose-it style of analysis of development. You get rid of it, you find out what happens to the organism. If something happens to the organism, then you conclude what? If you get rid of a gene and then development changes,
It's important for the development of the organ.
there's a specific term, so that suggests that it's important for development. If you get rid of the gene and that's all you've done, the only way you've tinkered with the organism, you got rid of one gene, and something changes, right, then that gene is important in that process. So, that means that the gene, and this is nomenclature that some of you have heard before, that means that gene is necessary for whatever process it is. Necessary. There are two contrasting types of experiments we're going to put up here. If you get rid of a gene and something changes, that means the gene was necessary for normal development. Necessary. Essential. You have to have that gene to do normal development. If you get rid of just one gene, that screws up development, then that gene was necessary to do normal development. The complement, so I'll often ask, and I will today or next time we meet, ask, what were the experiments that were done in the paper that we were reading? Were they necessity tests? Did they get rid of a gene? Did they knock it out, knock it down, lower its expression? Or did they test sufficiency of a gene to do something? And this is an experiment called a MOVE-IT experiment. What that means is there are two ways to test the role of a gene. You get rid of it to find out if it's important for a developmental process. But then if you have a hypothesis like, well, I didn't show you a picture of this, but I mentioned the Antennapedia fruit flies that grow legs on their antennae instead of antennae. So if you think that you have a gene that's responsible for leg development in fruit flies, and you want to know if it's sufficient for leg development, what does sufficient mean? If something is sufficient,
It's enough by
It's-
itself.
enough by itself to do something. So if there's one gene that's sufficient for leg growth, that means you should be able to move it somewhere else in the body, by which I don't mean physically move the gene, because every cell has every gene in the genome, but to change the expression. Normally, the gene Antennapedia is expressed in the parts of the fly that are supposed to grow legs. It's not expressed in the stomach, right? Why would you turn on a leg gene in the stomach? So the stomach of the fruit fly doesn't normally turn on Antennapedia, the gene. So an example of this MOVE-IT, or sufficiency test experiment, would be you turn on expression of that gene, Antennapedia, in the antenna. So somehow, and geneticists can do this in various organisms, we won't go into details yet, but we'll get there at the end of the semester. They can actually trick, in other words, the cells in the antenna to turn on Antennapedia, and then they turn into legs. So that's an example of a MOVE-IT experiment. You change where the gene is turned on in the body. So you move it elsewhere in the body. Or you can also move it in time. So you can fiddle with when during development a gene is turned on or turned off and see how that affects development. So I guess the best way to put that is move it in 4 dimensions. The 3 dimensions of the body, and then the 4th dimension, time. So where and when is the gene expressed? If you put it somewhere else, or if you make it expressed at a different point, do you still get the same sort of developmental process? What did they do in this paper?
They had mutants.
They did have mutants, but which kind of mutant? Were they changing expression of a gene, or were they moving where a gene is expressed? Wouldn't you know?
They were getting rid of them.
Sorry. No one ever calls me. Especially +1 0 0 0 0 0 0 0 0 0 0. Okay. So what did they do?
They were knocking out genes, they were knocking out Hox and Hox11 genes.
They were getting rid of
Rid of them.
HOX10 and HOX11 genes. So they were doing necessity tests. They were saying, what happens when we get rid of a gene? Okay. So, more relevant to the background of the paper, a little bit. That was all big picture background. Now we're talking specifically about this paper. Which genes were they working with? You just said. HOX10 and 11. So this is a picture of the different paralogous groups. We're going to talk about what paralogs are, because I didn't introduce that terminology either, and that's important for this paper. So a paralog is a gene that is the same gene. Go ahead. It's derived from a common ancestor. Basically, it means it's the same gene in multiple copies in the same genome. So these 3 rather, copies of HOX1, A, B, and D are paralogs. There's the same gene, they're just in different spots in the one organism's genome. So the 3 of those, how many paralogs of HOX9 are there in mouse?
4.
Yeah, right there. 4. So those are the situations where you expect redundancy. If you've got 4 copies of the same gene, if they all still do the same thing, then you should be fine. And then in zebrafish, there are other numbers as well. So the authors mention these 4 different paralogous groups in the introduction. They say, second sentence, 39 genes resides on 4 separate linkage groups, chromosomes in other words, designated A, B, C, and D. So that's what that is, A, B, C, and D. That's what they mean. There are, in mice, these HOX genes that are essential for development. There are between, depending on which gene you look at, 2 to 4 paralogs of each of these genes. In other words, there's lots of redundancy in mice for these presumably essential genes.
Is that what redundancy means? Because that was a question I was thinking.
Yeah.
I mean, because at the end or one of the parts they mentioned, like a complicated things of the HOX mutation retain considerable functional redundancy.
Functional redundancy, right. So that basically means, that's one of the questions, and we'll get there, but that's basically what they wanted to know in this paper, was if you've got 4 copies of a HOX gene, how many copies do you need to do normal development? Are they redundant? So how would you test redundancy? What would be the prediction if you've got 4 copies of, they're not working on HOX9, but let's pick on HOX9, since that's not relevant to the paper directly. If you have 4 copies of HOX9, what would you predict would happen if you got rid of one of those 4 copies? Just naive prediction, don't worry about details. Would you be normal development or messed up?
Normal.
Okay, normal. What happens if you get rid of 2 of the 4?
Normal.
Normal. What happens if you get rid of 3 of the 4?
Well,
Stick to your guns.
no.
Normal. Oh, okay. Well, it depends, right? So that is the question of this paper, is mice have multiple copies of the Hox genes. How redundant are they? How many copies of, in this case, Hox 10 and 11, how many normal, good copies of those genes do you need to do normal development? Okay, let's go to the punchline. What did they find? And then we'll go through the details of the paper. What's the answer to that question?
Well, when they mutated 3 copies and then 5 copies, it all messed up their development. Some of them lost lumbar vertebrae, the others lost sacral vertebrae.
Okay, so it changed development in various ways.
That the paralogs were needed for developing that line. So yeah, I think all of them. I think that's what.
Yeah, so we'll see as we go through the individual figures how exactly this works, but the take-home message really is that most of the paralogs have some sort of role in development. If you get rid of 1, you mess up development a little bit. If you get rid of 2, you mess it up even more. Some of the paralogs are more important for specific parts of animal development, but not others. So it's a discovery-based project. Was there a hypothesis in here? That's one of my favorite questions to ask, is it at the start. Is it a hypothesis-based project? Did they go in with an expectation? Did they state somewhere in the early part of the paper, we hypothesize that, or we think that, or we expect that this will happen?
I guess it was their title that they're required to globally comment.
Well, that's, the title is, Leslie?
Sorry, they did an observation. They suggested an observation at the beginning, on the first page.
So what did they say?
This observation suggests that Hox genes play a critical role in global patterning of vertebrate axial skeleton.
Mm-hmm. That's,
Based on like previous.
yeah, based on previous data. So that's more like the background reading that led them to do this experiment. We think that, based on previous research, the Hox genes are important in axial development. They're already mostly known. The degree to which redundant Hox genes, or potentially redundant Hox genes, paralogs, are important was not known. And so the title winds up being not so much a prediction, but the conclusion. And titles usually are. Titles of these Evo-Devo papers, as a lot of biology science papers, are the punchline. This is what we learned. That's how the authors get people to read their papers. Here's what we found. Come see the details. Okay. And the main reason I wanted to point this out now, this pattern of Hox gene presence, is I kept wondering, did anybody else I should ask? I don't know. If I ask the question, I'm going to give away the answer. How many different groups are there? There's 4, right? A, B, C, and D. 4 paralog groups. Which ones did they study? A, C, and D. So for example, well, we'll get there in a second. But yes, that's the answer. They studied A, C, and D. And I kept thinking, why did they study B? They said there's 4 linkage groups. That's like the first or second sentence in the paper. There are 4 paralog groups. And they never, and I kept looking and looking, and they never explained, what, why are you looking at, why not? Why didn't they study the 4th paralog group for Hox 10 and 11? Because I smacked myself in the head.
It doesn't
Realize-
have that.
Those have already been lost in mice. There is no Hox 10 or Hox 11 in the B paralog group. They could have just told me, but instead I wasted like an hour trying to figure out why they weren't studying the Hox B cluster. So that makes it actually easier for them to do the study with this group because they have fewer genes to try to knock out. It might have even been better for them to study like Hox 7 or Hox 2, which only already have 2 genes there. But on the other hand, then they wouldn't be studying how duplicate genes affect development, because if you study something like Hox 7 or Hox 2, you're back to diploid. You're now studying, you've got 2 copies of a gene. If you get rid of one, what's going to happen in development. And that's something you can do with any organism, not just mice. Okay. And I've already made this point. The point of this slide was a big difficulty for a really long time in EvoDevo, and it still is true, is you don't really, it's hard to tell what the effect of a single gene is if you do a lose it type experiment. You knock it out and you see what happens to development because of redundancy. And that's the reason that this is a really outstanding paper that actually made it into a very prestigious journal, Science. Because it's so technically hard to do this sort of experiment where you get rid of 1 copy or 2 copies or 3 copies or all 4 copies of a particular gene. In this case, 3 copies, but in theory, if you did it with one of the other Hox genes, you could do getting rid of all 4 genes.
I had a question regarding the 5 allele mutants.
Please do.
So, those 3, the A, C, D were the ones they were studying, but then for each of those, they were studying their alleles also. 5 alleles.
So you're gonna throw more jargon into the
Yeah.
discussion. No, that's totally fine. Which were the 5 allele?
Yeah, there was a five.
Was it for the Hox 10 5 allele mutants?
Which figure was that? I remember what you're talking about. I just can't find it at the moment. Oh, it's like 1P and 1Q. Okay. Well, let's see where I put that. Let's see. So we'll skip a bit, but what I would normally ask, good question. What I would normally lead so that was all background. That was nothing to do, that's what I would normally do on a Monday. This is where the presentation would normally start. I would start a discussion of a paper with big picture. This is actually something we just now talked about. What was the intellectual need? What was the hurdle? What was the barrier? What was the gap in knowledge? What didn't the scientists know when they started the project? What was the point of what they were doing? That's a good big picture way to start a paper presentation. After you give any more background that you think that you need for the paper that I didn't provide the meeting prior. So what was the big question? And why was it a big question? What was the state and knowledge of the field such that this particular question was not yet known? Or the answer to the question was not yet known.
I think it was to learn the function of the entire groups of both Hox 10 and 11 on the pattern to the development of it.
So that's a very specific, in response to this question, that's a very specific answer, is that yes, at this point nobody, and it's true, you're not wrong, nobody knew what the roles of individual Hox 10 and Hox 11 paralogs were. What a magnitude of effect does each paralog have in development?
They wanted to know when there's redundancy and when there are many copies of a gene, what happens if you mutate some of them, get rid of some of them? What happens to development?
Right, so that's a more specific need, which is nobody knows in any organism, that was sort of the bigger need. Nobody has yet known in any organism what the effect of redundant genes are in a very basic developmental process like axial patterning, deciding where you are in the head to tail axis. Okay.
They also look at it from an evolutionary aspect too, because they talk about how an absence or presence of these Hox 10 and 11 genes could have like promoted longer vertebrae. That could have increased flexibility, couldn't have increased flexibility. So.
Sure. So there are a spectrum of possible outcomes to this sort of study. The 2 extremes are you get rid of all but 1 paralog. You get rid of, say, in this case, 2 out of the 3, and nothing happens. You get rid of the last one, and then everything goes to shit. The other possibility on the other extreme would be you get rid of 1 at a time, and there's sort of a linear alteration, linear in effect size. Like you get rid of 1 out of 3, you kind of mildly disrupt development. You get rid of 2 out of 3, you more severely disrupt development. And then you get rid of the 3rd one, and all of a sudden you can't develop anymore, and you don't actually get a live embryo. So that was something that nobody knew when they were going into this, really was, what is that pattern like? Is it just a smooth sort of slow decline as you get rid of more and more paralogs, you get worse and worse at development? Or is there a threshold? Is there a point at which you've gotten rid of too many paralogs and all of a sudden you have zero development anymore? And really good scientists, actually, I'm not, let's see, I shouldn't record this. I am recording, but I should cover the microphone. Good scientists, and I'm just going to claim that all of the papers we're reading this semester are from good scientists, should always put that, these should be hidden at least, if not really obvious, in the introduction to the manuscripts. The answers to all these questions. Did you test a hypothesis? Did you have a prediction or not? Were you just going to mess with development and observe what happened? Report what you saw. Discovery-based science. We're not going into it, we're going into it blind. We don't have any predictions, we're just going to see what happened. We're going to mess with some genes and see how we screw up organisms. And because this is a critical component of grants, when you write grants to get funding to do any sort of research, no matter what field you're in, chemistry, biology, physics, astronomy, earth and environmental science, right, you have to be very clear at describing both what are you going to do and why does it need to be done? Why is this relevant? And usually that relevance is tied directly to, we know all this stuff, but here's a question that's really like obvious that no one's ever tested before. That's usually how scientists will phrase this sort of answer, and it'll usually be in the introduction of a paper. And in this case, I underlined the very top of the second page, the first column. Over the past decade, loss-of-function studies of mice with single and even subsets of paralogous Hox gene mutations have shown, blah, 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 only relatively minor changes in skeletal phenotypes. So what they're saying is some people recently have knocked out one paralog or maybe two, and that's only had subtle effects. And then they go on to say, end that sentence, which is inconsistent with their proposed role as global regulators of axial skeletal patterning. So that's them saying, it's subtle, it's buried, it's not really obvious. They're saying somebody should go on and do more, knock out more copies of paralogs and see what happens. And then lastly, I think, last 2 points that are common. Again, I hesitate to say expect, but I kind of expect every presentation to start like this, also mentioning clearly the experimental objective. What was it they wanted to do, the authors? And that follows in this paper immediately after that last sentence I read. What is complicated, this also, that intervening sentence in the left column of the second page also makes that point about what's the big picture, what's missing. What has complicated the analysis of Hox gene mutations is that these genes have retained considerable redundancy between paralogous groups. That's the hurdle. So they mentioned, here's something we don't know. That was the first thing. Then they say, this is the problem. This is the obstacle that's been keeping anybody from knowing this. It's the amount of redundancy of the paralogous genes. There's so many of them. Up until this point, we haven't had the genetic tools to get rid of all those genes all at once. And then, therefore, we examined the effects of the loss of function of the entire group of HOX10 and HOX11 paralogous genes. So they state explicitly in one sentence, this is what we're going to do. You'll almost always find this in introductions of all the papers we're going to read. The authors will say, this is the unknown, the question we want to approach. This is why no one's been able to do it yet. And this is what we did. They'll have a 1 or 2 sentence at the very end of the introduction usually. There'll be a 1 or 2 sentence thing that says, this is what we did, this is what we found. 1 sentence each, maybe 2. It's the best place, I think, in a paper to start reading if you're not familiar with reading primary literature yet. It's usually the last paragraph of the introduction. If you read, it's almost the same as the abstract. Sometimes it's just a little bit more information. So not only should this formally be in a presentation of the paper, more importantly, from my perspective, take it or leave it, is these are questions you should be asking when you read the papers. that's really why I wanted to put them in the slides, not so much to tell you how to do a presentation, but these are the sorts of questions that are important for you to think about to understand the paper, to get to a level of understanding the paper that we can then come here and discuss the manuscript. So let's do one figure, and then we'll break, and then we'll pick the rest of the paper up next class. That means that you don't have another paper to read for next class. Next Wednesday, we'll finish this paper, and then I'll do my last sort of general intro to molecular and developmental genetics. Good. Oh, so what are we looking at? What is this? It's a mouse.
The Lombard sacrum,
Yep. So, okay, so we're looking at
the rib.
these are ribs. This is spine. And so we're only looking at bones. They've done something to the poor mouse to get rid of all the flesh and organs, and they've stained with probably alizarin red or some other stain that I don't know the name of, that stains bone. So you can easily see the bone. So we're looking at the back of the mouse, basically, the back half of the mouse. Lumbar, sacral vertebrae. Is this a normal mouse? And then what are these?
They've cut specific vertebrae. They're looking at the cut pieces.
So I'm just trying to help everybody understand, if you don't already, how these are visualized, how this picture relates to this picture.
Like the first one is the thoracic T12.
Yep, so that's T13, so that's T12. So how does that vertebra relate to this picture?
Cut it.
And they've cut it and they're looking at it from like front.
Cross.
From
Yeah, exactly, it's a cross, so it's like you're looking down the spine, you take that and you lay it down, right? So that's looking at it from that end of the mouse, or the head end of the mouse. Okay, so it's just another way to look at the morphology. It's a different perspective of the same bony structures. Okay, so why is this important? What kind of mouse is this?
It's a normal.
Okay, so how do you know it's a normal mouse?
Because it's the control.
Okay,
Oh, yeah.
right, okay.
And it doesn't have any other changes. Like they described the pattern, it's like it's always gonna have 13 ribs or 13 sections.
It's not missing anything.
Right. Yeah, I found it, that's one of the, I wasn't trained as a developmental biologist, I developed developmental biology. And when they mentioned, what was it, something like the recipe,
Yeah.
Oh, yeah.
the AP axis recipe for the vertebrae, I thought that was kind of interesting.
Actual.
Yeah. Okay, so let's look at, we've got 2 minutes, we'll look at, then we need to compare against the control. This is it, this is the last slide for the day. So we just saw the control mouse, and I've redesigned this figure a little bit so we can always compare one other panel of that figure with the control. So what happened? What's this mouse? What's the one on top? This mouse 10A-ACCDD. So it's a triple mutant. It's got 3 mutations. In our nomenclature, then the number is the gene, okay, right? This is HOX10. They're studying the effect of, and what they're looking at is a mouse. What does AACCDD mean? That's in formal genetic nomenclature, little a over little a, little b over little b, little c over little c. So they're homozygous mutant for each of the 3 paralogs of HOX10. In other words, this mouse, its ancestors started with wild type, wild type, wild type, wild type, wild type, wild type. It had 3 different HOX paralogs, and in each of those, both copies of the chromosome had the normal version of the gene. So now they've gotten rid of 6 out of 6 of the normal versions of this gene. All of them. So it's called a triple mutant because they get rid of the 3 paralogs, but at the same time they've gotten rid of both copies of each of the 3 paralogs, right? From mom, from dad, from mom, from dad, from mom, from dad in a diploid organism. And what happened? Last point. You get rid of all 6 HOX10 paralogs, or all three, both copies, what was the effect?
They lost the longer ribs.
Okay.
Lots of ribs.
So they end
Lots of ribs.
lots of extra ribs. Yep. And they've lost some, well, they still have the lumbar region, but yeah, they've grown extra ribs. So, last question then. Is this, do you think, important to the mouse? Do you think the mouse, I don't know the answer, I'm just wondering. Is this important for development? Does the mouse really need HOX 10, A, B, and C? Does it matter if it's got little extra ribby bits on its?
It could probably damage its intestinal area, maybe it reaches it.
Yeah, I mean, right. It's more research to be done, perhaps. They didn't mention anything, of course, about, well, not of course. In my reading, they didn't mention anything about how these mouse were when they were alive. Mouse. That could have been kind of interesting too. Did they mention, okay, sorry, I'm a bad teacher because I keep getting excited and asking other questions. Next time, I'm gonna make sure this gets recorded so I can remember, we should ask the question, or maybe you can look it up for next class. What stage of development were these mice when they were sacrificed and stained? Are these embryonic mice? Were these adult mice? Because one of the expectations, and I'll leave you with this then, one of the expectations of developmental genes would be, well, if they're really important, then you don't even get born if you screw up development. It's embryonic lethal, in other words. At some point during embryonic development, development stops if you really screwed up development. So I don't know, if these were adult mice, that's okay. If these were adult mice, then we might conclude that, well, yeah, they might have had some internal organ issues, but if they could survive to adulthood, it's okay. But if the scientists never even bothered growing the mice to adulthood, we don't know the answer to that question yet. And that's why I thought of that. Cool. Any final questions? Concerns? Yes, sir.
So I was just wondering, have there ever been like any experiments of trying to add the genes back where it was missing, like that B that was missing? What would happen if you add that back and like see what happened there?
Right. And that's a perfect question to ask. And scientists do that sort of experiment. So if they find a mutant, for example, something that does weird development, and they think, oh, maybe this mutant mouse is missing HOXB, then they can put an extra normal copy of HOXB back and see if normal development is restored.
But what would happen if you just put in the B and took the others out?
Ah. There's something that could be done, but I don't know the answer to the question, of course. But yeah, that's exactly the sort of experiment that developmental biologists would do. Yeah.
So if you would like be interested in like how they actually did the mutation, like what were is there?
They've probably,
It seems like they just published the results. Right?
Oh, that's right. I was thinking about that, and then one of my kids came up and bugged me, and I forgot that was what I was doing at the time. So in Science and Nature, you can leave if you want to, I don't mind, we're done. In Science and Nature, because this journal has a limited number of citations you're allowed to put in your papers, I don't think it's actually 22, but it's like 25 maybe. Normally, materials and methods aren't published as part of the manuscript, they're published online as a supplemental something. Normally in Science and Nature, especially long ago, 2003, one of the references would be a look here for more information about our methods. Like they'd cite a different document. So this was before online supplemental materials. So I was looking for that, but I didn't see one. But one of the citations, 22, gives us no information, but they say, we are indebted to this one person for preparing some of the mutant mice. So some of the data, some of the materials and methods then we can't get information about because they didn't put it in the paper. And yeah, that's a major drawback of Science and Nature papers. But I would, yeah, that's basically where I would start is they either will cite somewhere in the paper, this is where the mice were created, and that'll be one of the many references. You could go look at that reference for the experimental details. I note that the Capecchi group has self-cited a lot. Most of the citations of the 25 are to this same research group. I mean, there aren't that many. There are a lot of mouse development labs, but there aren't enough. There aren't a ton. So they're basically just following up on their old work. So it looks like they probably published these mice earlier in some other publication and just didn't bother then giving that extra information here. Unfortunately. The best thing is to just contact the authors. Maybe I'll email Dr. Capecci and see if I can get a response by next class.